Hello, let's discuss on RNA. RNA is also called as ribonucleic acid. RNA is a single-stranded nucleic acid that is found in all the living cell, and it is the only macromolecule which functions as both storage of information and as a catalyst. So catalyst here referring to anything that is increasing or helping in carrying out the rate of reactions. So the RNA which is function as a catalyst is called as ribozymes. In case of RNA, the nitrogenous base thymine is replaced by uracil. So in RNA, thymine is not found, but instead uracil is found. So in DNA, the nitrogenous base are in the form of adenine, guanine, cytosine, and thymidine. So, and in case of RNA, it will be same for these three, that is adenine, guanine, and cytosine, but instead of thymine, as in case of DNA, it will be uracil in case of RNA. So this is the difference in the DNA and RNA. Now let's discuss on the types of RNA. RNA are of three types. The first one is messenger RNA or mRNA, also called as nuclear RNA. mRNA carries the genetic information as that of the DNA. In case of, as we know that in case of DNA, adenine always pair with thymine with 2 hydrogen bond, and guanine nitrogenous base always pair with cytosine with 3 hydrogen bond. So if we look at the DNA and RNA, if the DNA is at adenine, then mRNA will be uracil. That means adenine will pair with uracil in case of RNA. So instead of A equal A with T, in this case RNA, A will be with U, and for cytosine, for guanine and cytosine, it will be same. Now if this is the template DNA, when we say template DNA, we are referring to the strand of the DNA that is carrying the genetic information. This is the mRNA strand. So the arrangement of nitrogenous base will be in this form. If the DNA template have nitrogenous base A, G, C, T, C, then in mRNA it will be U with A instead of T. We know that A always pair with thymine, but in case of RNA, I told you that thymine is being replaced by uracil. So in this case, U with A, G, C with G, G with C, A with T, and G with C. So this will be the complementary strand of the mRNA. Now once the DNA template is copied, that means the genetic information in this DNA template is being copied by the mRNA, the mRNA will come out into the cytoplasms from the nucleus, where it will be getting attached to the ribosomes. Ribosomes is a cell organelle which is carrying out the process of photosynthesis. So the genetic information that is being copied or transcribed by this mRNA will be converted into a specific amino acid by these ribosomes. So this process of conversions will be carried out by the ribosomes, and many amino acids will be formed. So when the several type of amino acids are joined together, they give rise to what is called as the polypeptide chain. Peptide here refers to amino acid, and poly here refers to many. So many amino acid. Now let's discuss on the structure of tRNA, I mean mRNA. Structurally, the mRNA looks in this form. At the 5' end, the mRNA have methylated G cap. Now G here stand for guanine, and next to this is the non-coding region or the non-translated region. Now in this case, the non-coding region that is present between the methylated G cap and the initiation codon is called as the leader, and this is present just before the initiation codon. Now next to this non-coding segment is the initiation codon. This is the initiation codon, and in mRNA the initiation codons are AUG, GUG. When we say codon, we are referring to the triplet nitrogenous base. So next to this initiation codon is present the coding region from here till here, and just behind this coding region is present the termination codon. So termination codon in case of this mRNA is the UAA or UAG or UGA. So these are the triplet nitrogenous base which is signifying the termination codon, and just behind this termination codon is the non-coding regions, and just behind this non-coding region at the third dash end of the mRNA is the polyadenine tail, or also called as poly-A tail. So this is the structural components of the mRNA. Now next let's discuss on the second type of RNA, that is the transfer RNA, indicated by tRNA or adapter RNA. Now tRNA recognizes the codons on the mRNA, and it gets attached to the ribosomes for the protein synthesis, so to synthesize particular amino acids. So that is the function of tRNA, and tRNA is known by different name, also called soluble RNA or supernatant RNA or adapter RNA. Now let's discuss on the structure of the tRNA. The structure of tRNA is best explained by a model called as the clover leaf model, and according to this model the tRNA appear in this form. So according to clover and leaf model, the tRNA consists of 4 distinct arms. The first one is the amino acid arm or the acceptor arm. So this portion is called as the amino acid arm or the acceptor arm, and this amino acid arm consists of a 7 base pair possessing both 3' and 5', and at the 3 terminus the 3 nitrogenous base are present, that is CCA, and this 3 nitrogenous base at the 3' end can join with specific amino acid. Hence this end, CCA end at the 3' end, is also called as the amino acid acceptor arm or carrier arm. Now the next one is the DHU arm. When we say DHU, we are referring to dihydroxyuridine arm. So this portion is the DHU or the dihydroxyuridine arm. So this arm, that is DHU arm, has a stem of 4 base pair, and it ends with a loop. So this is a loop that is consisting of dihydroxyuridine base. So this is called as the DHU arm. And the third is the anticodon arm. This portion is the anticodon arm. This anticodon arm is lined opposite to the acceptor arm and has a 5 base pair, and it ends with the loop and this anticodon arm consists of a pseudouridine. Now the loop has 3 nitrogenous base pair, and this nitrogenous base pair is complementary to the triplet codons of the mRNA. Let's say this is the 3 nitrogenous base pair that is present in the loop of this anticodon arm. If this 3 nitrogenous base is coding for A, U, G, then the mRNA in this form will be U, A, C. So this portion will be carrying the nitrogenous base that is complementary to this triplet nitrogenous base. Now the fourth arm is the T sigma C arm. So this T sigma C arm has a site for attachment of ribosomes for protein synthesis, and this indicates the T sigma C arm, and the arm and the stem of this T sigma C arm has a base pair of 5. That means 5 base pair are found in this stem of the T sigma C arm. Now this T sigma C arm stand for T stand for ribothymidine, sigma stand for pseudouridine, and C stand for cytosine. So this is all about the clover and leaf model of tRNA.